So this is the review tutorial for cellular respiration and energy enzymes and all the things we've been doing leading up to cellular respiration and of course into the mitochondria. So this is our little test review to put all these major concepts together. So when we started this talk, we started talking about energy. And in our discussion of energy, we discovered that we're going to deal with basically two types. We're dealing with the kinetic form, and we're not going to talk too much about this, but that's the motion form of energy. And the motion form, okay, is when you see molecules move, they have kinetic energy. And a lot of what we're going to deal with, of course, is potential energy. Okay, and the potential energy, although not my favorite way to talk about it, is that stored version. And that's important to realize that we actually can essentially store energy as biological living creatures by essentially making making ATP, okay, phosphorylizing, or I should say hydrolyzing water to make ATP. Okay, so we get to do this. Now, how do we make ATP and why is ATP important? Well, adenosine triphosphate's got 3 phosphate groups and they have 3 negative charges in close, let's say, close area. And this creates high tension like a spring, very coiled. And by cleaving this one negative, or in this case, one phosphate off, energy is released. So what we need to understand is that in the body, we need to have a currency of energy. Somehow we're able to take ADP, adenosine diphosphate, and we've looked at these structures, and their structures aren't that important, but adenosine is a nitrogenous base, okay, and we have a ribose sugar attached to it along with some phosphate groups, but diphosphate means we have 2 phosphate groups. In any case, this molecule has only 2 phosphates plus another phosphate group. We could make ATP. So this is an important cycle. We talk about the energy we need to deliver to cellular functions. And those cellular functions could be building proteins, making collagen, building cytoskeletons, could be mitosis, meiosis. The living and breathing cell, it requires a tremendous amount of energy to do many different types of things. And you should understand the things that, or the things that we do is in something called metabolism. So in our metabolism, these are all of the chemical reactions that we control and regulate to provide life's functions. The understanding, though, is that when we do life's functions, whether building proteins, making enzymes, all the things we've talked about, to do and run the metabolism, it takes energy because a life or a cell, okay, to undergo mitosis and divide, okay, in order to reproduce, okay, you are essentially gathering resources from the environment to build something, especially when you're, let's say, making a protein. As we've learned already, we take amino acids, and amino acids are polypeptides. And what we do is we take these amino acids, as we've done before, and we put them together, other amino acids. And what we do, water comes off, and we make something called a polypeptide. So these amino acids come together to make a polypeptide. Now, a polypeptide, all right, is a what? Is a protein. And these proteins, because of their 3-dimensional tertiary structures, make the enzymes that are the what? The soldiers, okay, of our metabolism. What drives our metabolism to do cellular work or cellular functions are these enzymes. And they're making and controlling of them, regulated by operons and or regulatory genes, is something we've already talked about. But the important point here is that we need to take ADP and make it into ATP and then use that ATP to do the things that require energy. And you have to realize that building proteins, the example I make, requires ATP. Now, why is that? Why does that require ATP? Well, because of something about how the universe works. As we've learned two major laws of thermodynamics. One that we've already talked quickly, that the first law of thermodynamics was anything was possible, and energy was what? Conserved. We can convert what? Energy from one form to another. Okay, and that opens up the possibility that cells can just recycle the energies they use. Well, of course, that doesn't work because the second law of thermodynamics states that there's a direction of which energy flows. And we all know that energy flows from, let's say, hot to cold. And because it does that, that direction is driven by something called entropy. And entropy is an important term that helps us understand that the universe is entropy. We write this universe, okay, of the entropy, which is S, is always increasing in order for energy to what? Dissipate. Okay, so if you see a spontaneous process, and it's a word that's very important, spontaneous means what? That we are taking a process, a chemical process, okay, and that can be physical too, but mostly chemical in our body, and it is working, it is reacting, it is going in a certain direction because free energy is being released. And if free energy is being released, that means energy from a small area is dispersing out into the universe, and we would say the universe's entropy is increasing. Now, for those that forget, this entropy sometimes is good to understand as a measure of disorder. Okay, the best way to remember entropy is that it's the way to measure the flow of energy from a concentrated source to a diluted source. So if I have something called glucose, C6H12O6, which is a concentrated source of high energy electrons, as we've come to know, when that breaks down in the presence of oxygen into CO2 and water, we know that energy is, or free energy, is released. Okay, and this is called a catabolic event. Now, when we break things down, free energy is released, and this energy is free, is able to do work. So as we've talked about, we want to convert this what? ADP into ATP, and how do we do that? We do that essentially by taking in a fuel or a source or a nutrient, which we've learned to be proteins or lipids, fats, or in this case, carbohydrates, glucose, C6H12O6. If you think about how it was made by the what? The chloroplasts in plants. And they used what? The sun's energy from the what? From the environment to put it into the bonds, okay, so that glucose has high potential energy. So we break down this, or we use this fuel to do what? To drive ADP into ATP. And it's a spontaneous process because this molecule has a high concentration of energy. Or if you look at the reaction, a big molecule like glucose is breaking down into smaller pieces. So we can think of a big molecule like glucose breaking down into smaller pieces, and we can say that its energy is being dispersed into smaller pieces. And that's one way to think about it. But understand, entropy is about the dispersion of energy. Remember the Vsauce video we watched, okay? That was interesting to say that the only thing that we're going to leave behind is our heat that we've given off during our life as a warm-blooded individual, and the heat that we've given off to the universe has made the universe what? Even more chaotic or disordered. Okay, so let's graph this a little bit. Let's talk about what this means. So if I want to take my glucose and I'm going to break it down into CO2 and water, which is cellular respiration, I'm starting with a reactant, okay, which is my fuel, which is what? Stable, it has high energy, and I'm going to make my products. The products, of course, are the CO2 and water. And what we're going to find is the difference of where we start and finish, okay, because this over here is going to be a delta G, which is free energy. So the difference of where I start and finish here is our delta G. Now I really should say that this is free energy, okay, not the change. So this is the total free energy, and I say it's free because if something gives off energy, we can do work with it. Think about sitting on a pony. It's taking us somewhere. We're using the free energy the pony's given us. So if I burn glucose, gives off energy, I can do what? Boil some water. Okay, I can move a steam engine. We've been boiling water forever. So a fire that boils water makes steam. That steam can what? Move a locomotive. Okay, so there's free energy in reactants that have what? Concentrated sources of energy. All right. So in any case, we understand that delta G here is negative. Energy was given off in the process of taking CO2 and oxygen and making what? Glucose, C6H12O6, in the presence of oxygen and making what? CO2 and water. Delta G is negative, and negative means spontaneous. Spontaneous doesn't mean by itself, it just means that there's a what? There's a pathway. There's a way for this to go. Just like hot goes to cold, there's a pathway. The reaction works. Okay, look, you're starting high and you're going down. And you should recognize this. If you have a lot of energy, we call this what? Unstable. Think about that guy with the rap album in the city that wants you to buy it. He's got a lot of energy. So having a lot of energy is unstable. This is the energy of the reactants. It's high. This is the free energy of the products. It's low. How do we go from high to low? We must have what? Released it. And releasing something, we have a negative sign. So we started with a high amount. We went what? Down. And that's why delta G is negative. Now, if delta G is positive, the graph would look something like this. Okay, we'd be going up. Now, where in the world would we have a delta G going up? As I said, if this is the free energy, and I'm starting low and going high, okay, clearly this is going against the grain of the universe, because in order to start with a low amount of what? Energy, and go to a high amount of energy, I am going to need what? I am going to need energy to go into the process. There's no free energy here. This is like picking up the pony and now carrying the pony and providing work to go up the hill or move. Okay, so when does this happen? Well, it actually happens in the reverse. When we make, okay, sugar or glucose from the chloroplast of plant, they take in what? Energy from the sun. And they take the energy from the universe, and what they do is they concentrate it into this organic molecule called glucose. So they take CO2 and water, and they make with the energy from the sun. This is not a spontaneous process. It requires what? Energy from the sun. We make our glucose, or the carbohydrate, C6H12O6. So it's important you understand the delta G for this reaction is going to be positive because it takes energy. That means you need energy to make this happen. We have a couple of words to describe this. This process is called a what? Exergonic. What does that mean? That means we've released the free energy, started with a very unstable high energy and became stable. Exergonic reactions are always spontaneous, and that means that delta S, entropy, has increased. This one, because we're starting low and going high, is endergonic, I should say. Needs energy. All right? So very important. Now, if we think back to the cellular respiration, we start with the glucose, which is a concentrated source of energy, with the oxygen that we breathe, and we make these stable compounds, CO2 and water. Why do we do that? Well, we know this has energy. So what does it do? It releases free energy. We know that delta G in the reaction is negative. This free energy does what? Forces the ADP into ATP, and this is the energy currency of the cell. This is a high energy particle that stores a little bit of the energy that came from the glucose, and we can distribute it around the cell as needed, when we need it. So it's a currency because it's a smaller denomination. Glucose has tremendous amount of energy. If we were to burn it with regular oxygen, as I showed you with demonstration, it would burn holes in our stomach. It would kill cells. So if we just take a little bit of energy from it over time and convert it into ATP, it's a more effective and more efficient way to use the energy. So this is a what? Exergonic spontaneous process to go from glucose into ATP. That's what cellular respiration does. Now, what do we do with the ATP? Well, we use the ATP, who has the energy now, to force what? Cellular functions or cellular work. We call this using a what? An exergonic reaction to force an endergonic reaction, and this is very important. This is called energy coupling. This is what the body does. It takes something that spontaneously breaks down. In the case of glucose, the top one here, spontaneously breaks down into and gives off free energy. We tap that free energy, and we use that free energy to make something who is not spontaneous, which is ADP, into AT. So we're forcing ADP into ATP. So essentially, we're storing energy from the glucose into the ATP. And we're doing so in small increments because we're going to need this energy to what? Build proteins, build enzymes, make the cytoskeleton, divide the cell. All of those functions are non-spontaneous. Think how organized the cell. Think about what delta S is, entropy. Entropy tells us about the flow of energy from a what? High concentration to a low. When we build the cell, what we're doing is what? We're taking resources from the environment and we're putting them into a small area in an organized state. We're going against the grain as we do these functions. So in order to couple something that is what? Non-spontaneous and a positive delta G, we do so with something that has a delta G of negative. Coupling means we take an exergonic process that can drive an endergonic process, a spontaneous process where the delta S is increasing to do what? To force a process where the delta S is decreasing. That has to be absolutely understood. That's what the body does. If there was no energy coupling, there would be no life because all of life's functions require work or energy. In order for the cell to do anything, it requires its tremendous amount of energy, and we do so and get that from the food. Now, okay, great. Now that we have the basics down, spontaneity and the rate of reaction do not go hand in hand. As you learned from the catalase lab, okay, these enzymes that we make endergonically or exergonically, the enzymes that we make that are coded for in the DNA that get unzipped by the what? By enzymes to make messenger RNA, okay, that go out of the nucleus, that go into a ribosome, okay, to tRNAs have to what? Use enzymes to attach amino acids. All of that takes tremendous amount of energy, okay? That is not spontaneous. So we need these enzymes to make the reactions more efficient. Remember, as I keep telling you, the rate of reaction, how fast the reaction, has nothing to do with what? How spontaneous a reaction is. So if we think about this reactant, the glucose, has tremendous energy. and we want to make products. If we didn't have enzymes, it would react so slow that there would be not enough energy for the reaction to go. We saw an example of hydrogen peroxide, okay, H2O2. It happens by itself. It becomes oxygen and water vapor, but it goes so slow that it takes a long time for this to go. Okay, we produce H2O2 in our cells naturally, and this is a toxin, and we get it out by something called catalase. But we need the catalase to speed up the reaction. So even though something may be very spontaneous, which means a high negative number, okay, it doesn't mean it happens fast. So in a chemical reaction, we have something called a pathway. We have a diagram here. And I'm going to show you the pathway that the reaction takes. Now, it's important that you understand the pieces to this. Okay, this represents the activated complex. That's the part where the two particles, let's say oxygen and, or in this case, two hydrogen peroxides, okay, are about to collide and bond. They're overcoming their outer electron forces that are on the outside of atoms. So this is the maximum energy that you have to overcome for the reaction to finally to bond. This is the energy that we start with. So you should see already, okay, that this energy from the top of the hill down to here is the activation energy, sometimes called EA. All reactions require that. So to start a reaction, you need some. If not, papers on a desk will be burning already. So what a catalyst does, party people, it lowers this hump, this energy barrier that prevents reactions from going. And it does so by helping the orientation. Remember, it bonds either the induce fit hypothesis or the lock and key method. But what it does is it lowers this hump. As I like to say, it medicates the hump. So what a catalyst does is makes the activation energy smaller, which once was this value here, now is a smaller value. If you need less energy to start the reaction, the same available energy in terms of thermal energy will make the reaction go faster, as we saw in the catalase lab. So enzymes are an important component when it comes to metabolism because they make the rate of reaction faster. Without them, there is no metabolism to any extent that we could use. So in the course of a cellular respiration, where we're breaking down glucose into water and getting that free energy to make ATP so that we can run cellular functions, which are what? Non-spontaneous. A lot going on there, I know. In that process, what? That process, okay, we need enzymes to force this along. So even though this is a spontaneous process, because of the energy barrier that the reaction has, we need that enzyme to lower that activation energy, that barrier, low enough for it to go at a capriciable speed so that we can actually utilize the energy that's released. Okay, very important. Our entire metabolism is based upon the working function of enzymes. Now, of course, enzymes are these proteins that have a tertiary structure, a three-dimensional structure, and they have these active sites or these places due to their protein folding based on the what? The R groups. Remember that? Everything's connected. These R groups attach and fold from these amino acids and make something called an active site so that substrate can fit in, and by fitting in that three-dimensional area, okay, it contorses or it makes the protein, or in this case the enzyme, which is a protein, change its shape to help break something down or help put together. It's the three-dimensional shape that helps with the orientation, okay, of the reaction. Remember, for a reaction to occur, they have to collide, and they have to collide with the proper orientation in order to do so. Catalysts help with the orientation by pre-bonding with them and maybe exposing their active sites. Okay, and understand that catalysts have these coenzymes that might be sometimes called vitamins that help make the structure stable. They also can have cofactors, and these are normally minerals or elements that also can add to the stability and the shape. Okay, and that's why we need to have these vitamins. But back to my graph here, let's go over all the parts that you have to know, and let's clean this up to a new graph as I have way too much going on, but I'm liking it. All right, I hope you're liking it too. So, looking at another graph, okay, of energy, time coordinate. Okay, what do we have? Well, we have, let's say, a spontaneous process, high to low, exergonic, and here's our pathway. All right, well, we know that a enzyme, which is a catalyst, lowers the activation energy. So we have the activation energy of the uncatalyzed reaction, and then we have the activation energy of the catalyzed. We have the energy of the reactants. We have the energy of the products. Notice the products have less energy than the reactants. Why? Because energy was what? Released. Free energy was released if it's an exergonic reaction. This, of course, right here is the activated complex. All right, and probably the most important one is your delta G. Okay, difference of where you start and where you finish. This is delta G, and of course it's negative because I'm going down. All right, and we as a cell get this free energy by catabolizing, breaking down stored concentration energy from food, in particular glucose. All right, so that's important that you have those pieces down. Now, as we talk about delta G can be negative when it's what? Spontaneous. Delta G can be positive if it what? Needs energy to push the pony up the hill. But when does delta G become zero? And that, my friends, is when no work's being done. This means that we are what? Using the energy to do work. This means we're adding energy to do work. And this is no work is being done. Nothing is really being done. And the reason for that is because we're at what? We're at equilibrium. And that's important that you understand. Equilibrium is bad for the cell. Okay? We do not want equilibrium. We want to take our food, okay, and we want to what? Metabolize it into energy, ATP. And we want to keep the reaction going forward, so we have enzymes to what? Control and speed up the reaction. Now, if the rate that we're breaking down food is equal to the rate that we go in the reverse and put a double arrow here, we're not overall making any energy and the cell is dead. We want food to come in, make ATP, that ATP drives cellular function. So we want a direction to go one way. We do not want a process where A is going to B as fast as B is going to A. That means we're not really using up the food and that we're not really making any amount of significant energy. Important you understand that, okay? Equilibrium is the death of a cell. And I remember doing that demonstration of the tanks. Equilibrium is where? Not when the amount of reactants and products are equal. It's when they stayed constant. Stayed constant. Why? Because the rate of water I was pouring from one tank was equal to the rate of water in another. So it doesn't mean nothing's going on, but it means that there's overall no net change. Okay, very important. Okay, so we've talked about the major things here. Let's get specifics into cellular respiration. Now, there isn't big specifics, except that you should understand the big picture here. So we have a cell, this is a cell membrane, and in the cytoplasm, we have basically glucose, okay, is transported in from the bloodstream. Say we have a human cell, I guess, and we have glucose, and then in the process of glycolysis, now sometimes they call it anaerobic respiration, so glycolysis, okay, I didn't want to spell that, I've had a bad week of spelling, okay, so in glycolysis, this is very important. This is an anaerobic process. All right, anaerobic. Now, what does that mean, anaerobic? All right, it means that this is a place where we do not have to have what? Oxygen. So this is anaerobic. I forgot the O. Okay. Now, what happens in glycolysis? Well, we take the glucose, very simple. We make an overall net of a little amount of ATP, not much. We take the energy that's stored in what? The glucose, and we break this down into two pyruvates, okay? And in that process, overall 2 ATP are made. Now, I'm not asking for that kind of specifics, but in glycolysis, you should understand that we are collecting energy. We are getting energy without oxygen. And we are not where? We are not inside the mitochondria. All right? So what's nice about this is a quick way to get energy. It's the first energy system of anaerobic, sometimes called glycolysis. So we don't need to have oxygen. So we can gain some energy. But this is far and few between compared to the energy that this X bacteria called mitochondria can produce in its inner cellular folds. Okay? So we can get some energy without the oxygen. It's important. So it's not always oxygen has to be present. And this is a fast way to generate energy, but it's very inefficient. Any case, that's the main point. Now that in glycolysis, after making pyruvate, okay, and where do we go with it? Now the pyruvate gets shuttled into the mitochondria, okay, open pore. And immediately we call it the link reaction, okay, but we make acetyl-CoA. Okay, and we make two of them. And it's not important that you even know that, but acetyl-CoA is probably something you should be aware of. Okay, and this CoA is part of the enzyme that actually helps this along is vitamin A. Okay, vitamin B, excuse me. Now, once we make this compound, we go into something called the Krebs cycle or the TCA cycle. And for every one glucose, we make 2 pyruvate. All right, 2 acetyl-CoA. So we basically have 2 rounds of this cycle. Now, in the Krebs cycle, what are we doing? Well, we are making some ATP, no doubt, not a lot, a little bit. But what we're really doing is basically using these carriers, okay? We have NAD and FAD basically pulling these electrons along. So we have the NADH, NAD plus, and then we have the FAD, FADH2. So I can do this, H2. So the NAD and FAD are carriers. So what we actually are doing in the mitochondria, we're actually stripping out some high energy electrons that are carried by the NAD and the FAD. I'll just keep it like that. So these guys at different points are stripping away electrons. Now what they're really doing is taking the electrons, they're being held between a carbon and hydrogen, and as you've talked about, they're being held loosely because hydrogen and carbon have about the same attractive force. So these electrons are pretty free. They have high energy. Okay? Now this high energy electron is eventually shuttled to the electron transport system, which we've talked about being in the inner fold of the mitochondria. All right? But what we're doing, and now understand this, we're also pulling some electrons in glycolysis from the NAD. So we have these carrier molecules that are holding on to these high energy electrons, and it starts in glycolysis where these carriers pick up these high energy electrons. So they're high energy because they're freer to move between these two atoms. And in the carrier portion of these molecules or these proteins slash nitrogenous bases slash enzymes, they remain in that state until they are shuttled in to electron transport system. So the Krebs cycle is basically stripping away carbon and hydrogens, and you're making CO2, and it's where you're making most of your CO2, okay? And you are making some of the ATP. So understand our CO2 is made, okay, in the link reaction in the mitochondria, okay, where we're taking the pyruvate into acetyl-CoA, and also in the Krebs cycle. But this Krebs cycle is a constant working of stripping away these electrons from carbon-hydrogen bonds. Notice the carbon will wind up being CO2, and this will wind up becoming an H plus through that process, and we grab these electrons through the carrier. So the glycolysis makes some quick ATP, and it's there to make sure that we can quickly get some energy if we need it. Okay, if oxygen isn't in high enough quantities, and if oxygen is present, we drive this TCA cycle in the mitochondria and eventually the electron transport system. All right, and of course, you can call this oxidation phosphorylation, and it's important you realize that that happens at these inner membranes or these inside membranes where we have these protein complexes, which by the way are coded for by the mitochondria's own what? Plasmid or DNA, which makes it so efficient. Its DNA is always about it growing, making proteins, so it doesn't need the nucleus to help it divide, all right, as we've talked about at length. Now, what's happening in the electron transport system that we've talked about, and this is so vital, we're going to finally take these carrier electrons, these electrons that are free, and we're going to bring them to the membrane. I'm just going to make this bigger. And there's some really, really cool specifics that you can get into at higher level biology courses. But essentially, these high energy electrons carried in from the NAD and FADs, okay, and they do come in at different points, but that's not important. The FAD would come here, and the NAD, of course, a little later. It's not important that you know those specifics. However, these electrons, high energy electrons, get carried. And what they do is they essentially go down in energy as they're carried from one to the other. Why do they go down in energy? Because some of their high energy free movement is used to essentially push, okay, with a piston, okay, H pluses that result, okay, from what? Pulling the H and the C carbons away. Remember, we take the C from the glucose and many parts of this, and we're left with H pluses. So we've got a buildup of them. So what the energy does is as we pass around the high energy electrons from complex to complex, there's some cytochromes in there, and there's a lot of different complex things. It's pretty interesting. These are actually pumps. So the energy from these free electrons are used to drive these H pluses into the what? Inside membrane here, this interstitial membrane. So the H pluses are building up. As I said in class, it's as if pushing the water upstream or up against what? Up against a beach, the water's going to run back. So we're building up the concentration of H plus here. And these are like little pistons, unbelievable little machines that are using the energy of the electrons to pump these protons. And that is so very important, party people, because by increasing the concentration of these protons, what you are doing is creating a concentration gradient. So all of these H pluses, kind of like water that you're pushing out from the lake onto the shore, once they're run back. And we have this very important protein slash complex slash enzyme all attached in one. And we call this ATP synthase. And it's got a rotor on it and has a small channel that allows these H pluses to run back from high concentration to low. When they run back, they spin this rotor, which is the kinetic energy that's used to convert ADP that we've talked about into what? ATP. And although we're making, I don't know, about 2 ATP in the Krebs cycle and some ATP in glycolysis, most of all the other ATP, okay, 30 or so more ATP, come from the electron transport system because of, very important, this proton motor force. So if I ask you specifically what is making the ATP, all right, you could say, well, it's essentially the oxidation or loss of electrons that these, let's say, carbons lose okay? But really the real answer is it's this concentration gradient called the proton motor force that is really giving the energy to the ATP synthase to turn this turbine, okay, and give it energy to make ATP. So certainly isn't a very clear pathway, but understand the redox that we've talked about, the oxidation. It's sort of like oxidation. We're starting with electrons that are freely held. We're going to wind up at the end having oxygen, which is, we've learned, very small, loves to attract electrons, attracts its own electrons and attracts others. And this attractive force is pulling these electrons forward. Its attraction is pulling the electrons from one complex to another, which drives and pushes the H pluses out. Without the oxygen, you do not have any oxidation phosphorylation. Okay, you're not going to phosphorylate ADP, I should have that there, to ATP. We're not going to add that phosphate. All right, so the oxygen's needed to drive that, and when the electrons finally meet to the O2 with the extra H pluses, what we make is water. And in a water molecule, as I'm going to draw here, the oxygen tightly holds on to those pairs of electrons that were once free. So it's as if electrons were lost by the carbon and gained by the oxygen. And that's our redox. And you should realize that Leo, the lion, says grr. Losing electrons, which is what happens in glucose in a roundabout way, it lost the electrons, and who gained them? The oxygen. So electrons got passed from the oxygen, from the glucose to the oxygen that is in the mitochondria. We breathe for the mitochondria to do this. And it's important that you understand that this process is a slow burn to get a little bit of energy out at a time, because we want to make AT in great quantities, but we want to make stored amounts of small amount of energy. All right, so that's the bottom line, and hopefully that made some sense in a big picture of putting this all together. Hope that helped. Make sure you go to your textbook if you find something that you're still not sure about. Okay, 7 and 8 is what we believe focused on. Okay, go back to the worksheets. Okay, I'm asking for big understandings. Good luck.